Hi, everybody. Welcome to Micro Seminar. I'm Lizzie Wilbanks, and I'm one of the new co-hosts for the Micro Seminar series. And I'm really excited to be here hosting my first one. And our speaker today is Dr. Ellie Harrison, and she's going to be telling us about how the genome is alive and some really cool stuff about mobile genetic elements and bacterial evolution. So we're really looking forward to it. And I'll let Ellie take it away. Before I turn it over for questions, I'm going to be monitoring if you're in the Google Hangout with us here. We've got some more room. So the links are on our webpage and on Twitter. So you can ask us questions in here. You can hit us up with questions via Twitter, which I'll be monitoring. And then also, some new thing we're trying is adding in Etherpad. And so the link there is on Twitter. And so you can check that out and you should be able to ask questions that way too. So, all right. Thanks, Ellie, for joining us. And I'm going to let you take it away from here.
Thanks Lizzie, and thanks very much to anyone who's listening and for anyone who's seeing this in the future, mostly to the Lizzie and Dave who've run this fantastic seminar series. I encourage people to check out their websites. There's loads of wicked older seminars to catch up on too. Okay, so now I need to share my screen. Share. And start this up. So Lizzie, is that working? Okay, yeah, I'm assuming. Yeah, okay, great. Okay, so thank you very much for joining. So I'm Ellie Harrison. I'm a NERC fellow at the University of Sheffield, and I am fascinated by the world of mobile genetic elements. And I'm hoping that I can try and convince you that in order to understand these kinds of elements, we really need to be thinking about them as evolving entities. Okay, so mobile genetic elements are, of course, ubiquitous in microbial communities. I think we often tend to think of them as very much molecular machines, essentially, providing functions that are really important to bacteria. And this is certainly true, but they are also autonomous, or at least semi-autonomous elements with some control over their own replication and therefore their own evolutionary interests. And for this reason, I at least consider them as integral members of microbial communities. And I think perhaps the best analogy is to think of them as the endosymbionts of bacteria, which provide many functions to their host, but are kind of separate from what we consider the individual. So what am I referring to when I talk about mobile genetic elements? Well, the definition is kind of hazy and totally open to interpretation, but my 2 cents is that they are essentially any kind of units of genetic material that, unlike, say, a cell or a sort of life form, rely on the machinery, the cellular machinery of others, but have a transmission advantage over the rest of the genome. So for prokaryotes, this is all about having some route for horizontal transmission. And this separation, this sort of advantage, is really where all the magic happens, because while the rest of the genome has selection and acting on the genome as a whole, because they're inherited together into a new individual vertically, for any elements that are able to do a little bit of horizontal transmission, selection can then act in the best interest of that element, separate from that of the host. And yeah, sure, this is probably going to, for the most part, be the same thing, but it can set the stage for conflicts to occur between these elements and the host that they rely on. So this includes things like the phages. Now, these are obviously the perhaps the most autonomous. They can survive outside of the cell, but they do require cellular machinery in order to replicate themselves. Then inside bacteria, you have things like the conjugative plasmids, so extra chromosomal elements that can facilitate their own horizontal transmission, doing this through conjugation. And there are also many elements within the chromosome as well. So the sort of collection of intricative elements like the prophages, which are viruses that can integrate into the bacterial genome, as well as the sort of slightly murkier world of genomic islets, some of which are mobile and possibly related to phages. And then things like the ICEs, the snappily named integrative and conjugative elements, which probably are some relation to conjugative plasmids, but can do this conjugative form of transmission. But added to this, there's an extra, oh yeah, so they're kind of phage and they're kind of plasmid, sure. Added to this, there's an extra layer of mobile elements which can't necessarily control their own horizontal transmission, but can exploit that of others and therefore acquire this transmission advantage. So like phages which stow away in the viral capsules of others, the satellite phages, the mobilizable plasmids that aren't conjugative themselves, but can use those proteins produced by conjugative plasmids in order to move about. And things like transposons that aren't necessarily about finding a route for horizontal gene transfer, but more about moving between these chromosomes and therefore hitchhiking on the mobilization of others. And of course, these elements, apart from just being quirky and mad and interesting, are really important, playing major roles in bacterial populations. Of course, they are massive for vectors for horizontal gene transfer, carrying things like virulence factors and antibiotic resistance genes, things that we really, really care and worry about in our societies. They are major predators in these communities. So phages, of course, in particular, one of the major sources of mortality for bacteria, driving really strong selection on those populations, but can also be deployed by the bacterial hosts in the cases of temperate phage, the prophages that sit within bacterial chromosomes, and used as a form of weapon in bacterial warfare. So they can be sent off to go and kill competitors in the environment. And finally, they act like editors of bacterial genomes. By jumping in and out the bacterial chromosome, they are disrupting genes and are a source of essentially mutagenesis, which can greatly accelerate adaptation of the bacteria. But they also provide the kind of hard, what's the word? Sort of a testing ground, essentially, for producing new functions, because you have this very disposable sets of genes which aren't essential and can be co-opted by the bacteria for other things. And this has been true extensively within bacterial evolution. Many examples of secretion systems and so forth coming from phages, for example. So my interests really lie in trying to understand how evolution shapes these relationships, and therefore what impact this evolutionary understanding has for the functions that they provide. So for example, how does it affect the maintenance and transfer of the traits that these mobile elements shuttle around bacterial genomes? More recently, perhaps I've become interested in how these kinds of changes will ripple out to affect the wider community by altering the bacteria with which they interact. So the talk I will go through today is kind of looking at 3 different viewpoints on this. So the first 2 will span 2 big chunks of my postdoc work, working in Mike Brockhurst's lab at the University of Liverpool and later at the University of York. First up, I'm going to look at the role of evolution in bacteria-plasmid relationships. And I think this is a really nice example of how an evolutionary perspective can be really important to understanding the fate of these interactions. Then one of my favorite bits of work, I guess, was looking at how these interactions can affect other members of the bacterial community in quite simple environments. And finally, I'm going to give you a little sneaky peek at the work that I'm doing as part of my NERC fellowship, which I've only just started, so I really don't have any data, but I'm excited about it, so I'm going to tell you about it, basically. Okay. So to begin with, looking at bacteria-plasmid relationships, really a lot of the work I've done has revolved around a really simple observation about bacteria-plasmid interactions, and that is that when first acquired, plasmids are often incredibly costly to their bacterial hosts. And there can be lots of reasons for this, and I can recommend you the really great trends paper from Dave Baltrus's, I think actually it was just Dave's work, from 2013, that looks into these different sources of cost. Because I think we really began, at least I began my postdoc thinking of the cost of these elements as being very metabolic burden. So they're using up cellular resources and cellular machinery, occupying ribosomes, that kind of thing. And this is probably to a certain extent true. But I think what a lot of people are finding now is that the sources of these costs can be a lot more expensive. For instance, through disruption to cellular regulatory networks, or direct epistatic interactions between genes carried on the chromosome and genes carried on the plasmid, perhaps even just a single gene. But what a lot of people had found way before I even started my scientific career was that if you allow bacteria and plasmids to just evolve together in simple environments, we see that this cost becomes reduced. And this is essentially through compensatory evolution, ameliorating that cost of plasmid carriage, whatever source it might be from. And so we investigated this process of compensatory evolution, looking at a really nice model system. So I worked with a plasmid called PQBR103, catchy, right? Which was isolated and described by Andrew Lilly and colleagues from a beet field, sugar beet field in Oxfordshire. And what's great is that we also have a bacterial host taken from that same field, so taken essentially from the same community, and that's the Pseudomonas fluorescens strain SBW25, which I'm sure many microbiology type people will have heard of. And when we put these guys together, we find that the plasmid imposes quite a hefty cost on the bacteria in terms of fitness relative to a bacteria that doesn't have the plasmid. So this is about a sort of 50 to 30% reduction in fitness when these two are first paired together. And we are still unpicking the root of this cost, the mechanism that's driving this cost. But one thing that we can observe is in the transcriptome of our plasmid-carrying cells. So in these ancestral bacteria, this newly acquired relationship, we see a huge transcriptional response. So, I mean, this graph is a little complicated, but essentially you're looking up in this corner here, And each of these dots is a single bacterial gene. And I found that about a fifth of the bacterial genome is upregulated when the plasmid is first acquired. But this relationship isn't solely costly. Like many plasmids, the PQBR103 confers beneficial functions to its host in the form of carrying bacterial accessory genes that the bacteria can utilize under particular environments. And in this case, it's a mercury resistance operon. And so that means that when we increase the concentration of mercury in the environment, the fitness of the bacteria carrying the plasmid increases relative to that of the plasmid-free bacteria. And at very high concentrations, the plasmid is beneficial. It's almost essential because plasmid-free bacteria are dying through mercury toxicity. But I think what was really interesting is that when we looked at our transcriptional profile of these bacteria carrying a beneficial plasmid, so in this mercury environment, we really see exactly the same relationship. So this huge transcriptional response. And I think this really nicely shows that while the net effect of the plasmid for the bacteria might be beneficial, the source of the disruption that it causes to the cell is still there. So it's more a case of offsetting that cost than removing it. So to look at this in a bit more detail, I picked environments from this mercury gradient, so going from no mercury to very high mercury, and a number of intermediate environments, to look at how compensatory evolution occurs across this sort of spectrum from a very antagonistic relationship to a very beneficial mutualistic relationship. And I evolved these bacterium plasmids in these environments for about 450 generations, bacterial generations, and tracked the frequency of the bacteria throughout in my independent replicate. So this graph to the right here is just showing plasmid prevalence through time for those 6 different mercury environments, and each line is a different replicate population. And what we see is that the plasmid persists, and this is true obviously when there's lots of mercury in the environment, so when the plasmid is beneficial, but also in the treatment where the plasmid was very costly. So even though it has this huge, around 50% cost, and initially the frequency of the plasmid goes down very rapidly, it is maintained in all those populations. So, taking evolved clones from each of those replicate lines, I sequence the whole genome, plasmid and bacteria. This figure is kind of a summary of those genomes for each of my treatments. So from high mercury on the outside in red to no mercury, very costly environment, to the blue in the middle. And the dots are mutations, with the size of the dots showing the number of times that they occurred in my independently evolving lines. So what we can see is that there are several loci which were targeted repeatedly, both within treatments and across different treatments, and these evolved independently. And there are 2 loci which only evolved in the presence of the plasmid, so not in my plasmid-free lines, and these were the loci GAC-S and GAC-A. And together, these loci form a 2-component regulator of extracellular protein production. So, I mean, at first glance, this has nothing to do with plasmid carriage. But when we produce the knockout mutants, we see that while we have this, I'll use my mouse here, oh, there we go. While we see this, we have this big cost in the wild-type bacterial background, that cost is almost entirely ameliorated in the 2 knockouts of either of the gene. So we can clearly see that these are compensatory mutations which are ameliorating the cost of the plasmid, and that this is occurring whether the relationship was initially very costly and had no benefit whatsoever, up to where it is almost an essential element for bacterial survival. And I think what's quite nice is when we look back at the transcriptional response, we see that big sort of transcriptional freakout that we saw in the ancestral background. In the evolved lines, this gene expression response is almost completely resolved. And the evolved gene expression profiles really just looked like a bacteria that had never seen a plasmid in its life. So these are compensating for the cost of the plasmid by resolving this disruption. And I think I really like that we could actually get in and track some of these alleles as they moved through our populations. This is a really nice feature of these kind of experimental evolution studies. And we can see this is just one population taken from the no mercury environment. And the blue shape is the frequency of the plasmid over time in that population, and the orange is the frequency of the GAC mutation, in this case a GAC-S mutation. And you can see very nicely, I think, how the mutation arises and spreads into the population, basically rescuing the plasmid, which was on its way to extinction in that population. So this is it's great news if you like evolution. We could say that compensatory evolution can rescue plasmids that are on their way out. But I did some simulation modeling with Calvin Dytham, and perhaps intuitively, we showed that this very much depends on the rate at which these different events occur. So obviously, if plasmids go extinct before there's been time for compensatory mutations to arise and spread, then compensatory evolution is simply not going to rescue plasmids. But a prediction from this then is that this process of compensatory evolution is going to be more likely to occur, or at least be effective, when plasmid carriage is beneficial to the bacteria. Because under these environments, plasmids are going to be maintained at high frequencies and over a longer time period. And this gives a greater probability that mutations will spontaneously arise and spread, like so. In environments where the plasmid is costly or the relationship is antagonistic, we could say, plasmids may simply be outcompeted before these mutations arise. So in mutualistic environments, so environments where there's benefit to carrying the plasmid, we expect to see compensated, this sort of resolved relationship evolving. And so, in other words, we can describe these kind of environments as hotspots for compensatory evolution. And this, if we imagine that bacteria, of course, exist in very heterogeneous environments, they are not in these nice single environment lab cultures that we tend to look at them in. This means that dispersal of these compensated genotypes arising in our hotspots into environments where they're likely to, less likely to occur, can lead to plasmid prevalence being maintained, not just in that hotspot, but across a patchy environment. And so we wanted to just put together an experiment to test this, this idea. Okay, so I did this by setting up experimental environments where there are patches of no mercury on the left-hand side, where the plasmid is costly, and patches of high mercury, so those mutualistic hotspot environments with migration in between. Looking at the frequency of the plasmid in our antagonistic, our costly environment. As controls for this, we have homogeneous environments where our focal patch is antagonistic, but also our source patch, where our migrants are coming from, is also antagonistic, and the reverse, so where both are mutualistic environments. And I allowed for 3 different migration rates, so from very low migration to around 1% of the population being transferred. This is every transfer, so I think this experiment ran for 24 transfers, and 10%, so a significant proportion of the population moving into that focal patch every transfer. So if we look at the prevalence of the plasmid in those focal patches, so the ones on the left of my little schematics here. So in the absence of mercury in either patch, we see that plasmids were reduced to low frequency. Some were lost, but others stuck around for a bit longer. In our homogeneous but mutualistic environment, the plasmid is basically near fixation, it's beneficial. But in our mixed environment, where our focal population the plasmid is very costly and should otherwise be lost, we see that the frequency of the plasmid ranges basically from being very low to being near 100% of the population. And this very much depends on the frequency, the rate of migration into that population. But I mean, this seemed to prove our hypothesis, but what I found quite surprising, or perhaps, yeah, a little surprising, was that this wasn't necessarily determined by whether or not these GAC mutations arose. GAC mutations, which are here shown in the darker colours. So this is the frequency of the plasmid over time, and the grey shows the bacteria carrying the plasmid that were in these focal populations from the beginning, with the darker colour being the compensated bacteria. I can detect GAC mutants in all of these populations, so even in the absence of any benefit to plasmid carriage. But what I think is showing this this shows quite nicely is that what this is really being determined by is the population size of those compensated mutations. So here we see these blue bacteria coming in, and these are bacteria that have coming from the source patch. And regardless of mutation rate, we see them invading the population, but they never reach high frequency when the environment is costly. But in the heterogeneous environment, the prevalence of the plasmid is totally determined by these immigrant compensated bacteria, which are in this blue color. So here, whilst I think it's definitely true that the probability of these mutations occurring will range across these costly to beneficial environments. Here, they did occur, but by the time they occurred, they were at such low frequency that they're unable to increase or to produce a high rate of plasmids in the environment, whereas migration from populations that are beneficial and where there is a high population density of these plasmid-containing bacteria leads to almost complete plasmid prevalence across these landscapes. So, in short then, compensatory evolution is one way of stabilizing plasmid prevalence. And this will depend on a kind of race between these different competing processes. But if we consider heterogeneous environments, we can kind of understand, I think, much better why we see plasmids, which appear to be very costly, being ubiquitous across environments. Because if you have a subset of that environment being a place where the plasmid is beneficial, it can set the pace of compensatory evolution across environments more generally. Okay, so moving on, this next piece of work that I'm going to present is looking at how these kinds of evolutionary interactions can impact on other members of the community. So I have my bacteria plasmid relationship, which I've described previously, and we know a bit about what to expect from that because we've seen that evolution tends to resolve the cost of the plasmid through compensatory evolution. But added to this, I also have an extra predator in this system. So this is the bacteriophage Phi2, and the bacteria that I work with, SPW25, has a really strong history of looking at the antagonistic co-evolutionary interactions between this bacteria and this phage. And we know that it tends to take the form of a rapid arms race of resistance to the phage, driving selection for greater infectivity of the phage to infect the bacteria. So I wanted to know how these two evolutionary relationships with the same host, but otherwise separate, they have no direct interactions with each other, might shape the trajectory of evolution in this system. So to do this, I set up experimentally evolving communities. So I have my plasmid containing treatments, plasmid-free treatments, and phage-free and phage-containing treatments in sort of factorial design. And I evolved these sort of very simple communities for about 150 bacterial generations. So we have two interactions here, and so there's two kind of perspectives, ways of looking at these relationships. I'm going to start with the kind of phage eye view. So how does the presence of the plasmid impact this bacteria phage coevolution that I described? So the data that I'm going to break this graph down from the beginning, since it's a little hard to understand a bit. So I have my two phage containing treatments, so with and without the plasmid. And I took the bacteria and the phage that evolved from these populations and separated them out and compared or challenged the bacteria against the phage from phages from their own treatment and phages from the other plasmid-containing treatment. I did this just by streaking assays, so looking, scoring bacteria for phage infection. Oh, that's a lie, actually. I did that in a plate reader. So it's a quantitative estimate of phage infection. Okay, so this plot then is a reduction of something like 12,000 crosses of bacteria and phages, but essentially it's averaging out the susceptibility of the bacteria to phage infection. So firstly, we can compare those two bacterial populations, and we find that the bacteria taken from the plasmid-containing treatment had a much greater level of susceptibility to whichever of the phage populations that it was challenged with. Whereas overall, the susceptibility of our plasmid-free bacteria was much lower. So the presence of the plasmid has appeared to constrain the resistance evolution of their bacterial hosts. What's nice, though, is that we can also see the kind of knock-on impacts this has for the phages. So while in our plasmid-containing treatment, like the plasmids are just rubbish, they're no good at all. In the plasmid-free treatment, phages taken from the that same plasmid-free treatment were much more infective than phages from the plasmid-containing treatment. So obviously there's not a lot of selection to be a good infective agent when your host is very wimpy. But I also noticed something else about these populations, and that is that whilst in the absence of phages, we saw the kind of typical colony morphologies that you see for bacteria, for Pseudomonas bacteria. We also, in the presence of phages, had an extra colony morphology, and that's the mucoid morphology. And when I looked at the frequency of these mucoid colonies in our plasmid-containing treatment versus the plasmid-free treatment, we find that they're much higher frequency when the plasmid is present. And this is interesting because mucoidy is an alternative form of bacterial resistance to phages. So it's separate from the one that my assay is designed to measure, which was very much more the kind of all or nothing phage infectivity. This is a sort of a kind of slowing down of the rate at which phages are able to absorb onto the cell surface. So it's a different mechanism. And so what this suggests is that rather than my original statement that plasmids are preventing bacterial resistance to phages, it's more that they're leading them to opt for an alternative form of phage resistance. So flipping that around then, we can ask, how does the presence of the phage impact on the relationship between bacteria and plasmid? So here I was looking for compensatory evolution, which I've described as being a really important part of this, this evolutionary relationship. And we can track the frequency of these GAC mutations, these compensatory mutations in our populations through time, and find that when phages are present, the frequency of these compensatory mutations is far lower than it is when they are present. So phages are constraining that compensatory evolution process. So for both of these, then we can look at the changes that we observed in the genome by sequencing the endpoint mutations or the endpoint clones from this experiment. What we find is that we see quite a nice separation of the types of targets that were being hit across these different treatments. So this is sort of summarizing the loci that were targeted in the different replicates for each of those 4 treatments, finding kind of completely different mutations occurring when there's no plasmids or phages, kind of typical phage bacteria mutations when it's just the phage that they're having to deal with. So this is sort of LPS targets, so targeting things that stuck out on the cell surface. We see our compensatory mutations here occurring in the plasmid-only treatment. But in our tripartite treatment, very different mutations arise. So these are targeting genes involved in alginate synthesis, which underlies that mucoidy phenotype. So to summarize then, we find that simultaneous interactions lead to very different evolutionary trajectories for the bacteria. And I think this is a very simple example. This is a sort of one phage, one plasmid, one bacteria in a lab media system. But what it shows is that you can have very, very different outcomes just with these very small changes to your communities. So finally then, I think I have a few more minutes just to introduce the project that I am moving on to at the moment as part of my NERC fellowship. Basically, I haven't got much to show you other than to kind of plug it as something that people might be interested in, and I'm always happy to chat about it. So for this project, I'm moving from plasmids into looking at temperate phages, and these are really sort of the ultimate, I think, mobile genetic elements, because they're both phage and these kind of integrative elements that can be mutualistic to their bacterial hosts. And I'm looking at them in rhizobia, which, if you aren't familiar with them, are really important nitrogen fixers that live within these sort of root structures in legume roots. So these are root nodules, and form this really, really specialized mutualism where they provide the the plant host with nitrogen, fixing nitrogen and exchanging this for other nutrients that they need. But what's neat about these bacteria is while they have this really, really specialized relationship, they also spend a lot of time living freely in the soil. So when these nodules break down, they're just released into the soil community. And here they're going to be having interactions with many other members of the microbial community, including, of course, the prophages, or phages in general, in fact. And I've been having a look at prophages, for prophages in rhizobia genomes, and from my preliminary work, I've found that they are very abundant and quite variable. So, for instance, I've sequenced a couple that I isolated from one single strain, and there's a few phage in there, two genomes that I have really good sequences for, and one of them is a sort of mesorhizobium-like phage isolated from a very similar species, but another is most similar to one that is found in E. coli and Salmonella, so potentially coming in from very different communities. And what's nice about this system is that the bacteria then is interacting with two very important partners, with its temperate phages and its plant hosts. And both of those relationships are imposing strong selection on the population. So this is a phylogeny of rhizobia strains collected by Peter Young's lab at the University of York from just a single square meter of soil. And they're very diverse. This is essentially called one species, but essentially more like five. And the color of the dots is determined by whether they are specific to one host plant, clover, and the orange is the ones that are specific to another host, vetch. And then so the bacteria have this very different host range, and this, incidentally, is determined by a plasmid, so it's all good. But taking these phages that I sequenced and looking for their host range, we find that this host range is really spread across this population. So there are layers of host ranges determined by these different interactions that the rhizobia have. So my fellowship then is asking a couple of questions. Firstly, what what are these phages doing? We really don't know anything, or we know very little at least, about temperate phages in terrestrial environments. And then I'm going to get down into my evolutionary questions, looking at how the co-evolutionary relationship between the bacteria and the phage occurring outside the host, likely to be a sort of antagonistic interaction driving strong selection, might have impacts for the rhizobia legume interaction when they form. And the reverse, how does this very, very specialized and actually a mutualism that involves very strong bottlenecking, because each of those root nodules is formed by mostly a single bacteria, impact the mobile genetic elements that are interacting with the bacteria, the rhizobia. Okay, so final thing to do is then my acknowledgements. I've given these a little festive flair for you. So all of this, the work, all the data I presented was from Mike Brockhurst's lab, first at the University of Liverpool and later York. And I have some key collaborators who co-supervised my first postdoc, Andrew and Steve, and a huge number of people who are really instrumental in the work that I've presented. And yeah, thanks for listening. Okay, so now I need to get...
Great. Well, thank you, Ellie. Dave and I have been brainstorming. So I'll start us off here with some questions. I was wondering, it's so cool thinking about the influence that the plasmid and the phage had on choosing that capsular versus LPS mutation. Do you have any thoughts on like the mechanism of why the plasmid would tip the balance in one way or the other?
So actually, as pretty much all of my postdoc career, it comes back to this GAC relationship, right? So in that those gene expression results that I showed, we have some of the genes that control the biosynthesis of alginate. So they are upregulated. Now, what's funny is that I couldn't actually measure any difference. So I did some sort of colorific assays looking for a kind of increase in this mucoidy phenotype in the ancestors that have the plasmid versus the plasmid-free. And I couldn't measure anything. But the transcriptional response suggests that carrying the plasmid just increases the expression of those genes at least a little bit. And I have spoken to people who know a bit more about this pathway, and to them that sounds like something. It's a pathway that's quite complex, and you can have this priming where the early steps in the pathway are upregulated, but you don't get the phenotype until another transcriptional switching happens. So I think it's a case that these bacteria have primed the plasmids have primed the bacteria to evolve this phenotype. But what's super cool is that phenotype is also controlled by GAC. So what that means is that if you adopt mucoidy as your strategy for resisting phages, then you if you evolved the GAC mutation, it would be really costly because it would knock out that phenotype. So it's a kind of antagonistic pleiotropy situation. So whereas the GAC would be beneficial in the absence of phages, when the phages are present and you're relying on that phenotype, it's costly.
Cool. All right, well, we have a question here from Tyler Arbor. And Tyler says, I'd love if Ellie could expand a bit on the migration rate of plasmids in the population and how this is determined.
Well, so I mean, in my model system, I'm imagining more the migration rate of bacteria, right? So we know that bacterial migration rates, not from my work, from other people's work. If you think about like a soil environment, bacteria can easily migrate over scales of centimeters, and you have significant migration across much larger scales. So perhaps not the rates that I was looking at, but you can detect the same bacteria on the other side of a field, for example, like 150 meters. So if we think of bacterial migration in the scale of centimeters, and then consider the kind of environments that they're actually living in, which are heterogeneous on the scale of micrometers or smaller, you can kind of see that actually from a bacterial perspective, it's likely to be very unconstrained there.
Cool. Thank you. And then let's see, it looks like Dave had some questions too. Dave says, can you isolate phage from inside rhizobium nodules, and any hints of symbiosis related genes in these prophage?
The phages that I've isolated so far all come from bacteria that were isolated in nodules. So these are prophages which are associating with the bacteria during the symbiosis. In terms of free phage, I haven't, like lytic forms of phage, I haven't looked yet. It'll be really hard because these lysogens, these bacteria that carry prophage, do spit out a certain amount of the lytic form of the phage. It'll be quite hard to separate out those things. But I think the real kicker will be moving into the environment. I've done that thing where I like quite naively, I'm like, oh, I'm going to find loads of bacteria in the soil, and I know it's going to be pretty hard. But I think that's where I think a lot of really cool interactions are happening, but we know virtually nothing about what happens when they go into these soil communities.
Mm-hmm. Mm-hmm. Cool. And then Dave's second question was whether you've seen any hints of symbiosis-related genes on these prophage.
Oh, yeah. Well, okay, so it's like your typical mobile element, right? So there's, for the phages that I sequence, it's about 40% are known phage genes. So all of the required phage genes are identified in those genomes. The rest of the genome is all orphan or hypothetical or unknown function. So as I'm sure Dave has found looking at his plasmids, there's a huge amount of genetic material which could well be doing something, but we don't know anything about it. So unlike the plasmids, for example, where it's really clear that they are affecting the symbiosis, at the moment it's totally up for grabs.
All right, cool. Well, thanks so much. I think we're going to go off air here. So for all of you who tuned in, thanks for joining us. And I want to remind you, give a plug for Will Levitt's micro seminar that he'll be doing coming up 2 weeks from now in December. I think it's, I'm forgetting the date, but it'll be on Twitter. So stay tuned for our next broadcast later this month. All right, bye. Thanks, Ellie.
December 18th.
December 18th. Thanks, Dave. All right, thanks, everybody.